Hello students, now we'll start with new topic, that is second chapter of 11th class NCERT, Biological Classification. Monera are nothing but prokaryotes, which deals with all types of bacteria. Monera is nothing but considered as the first evolved kingdom according to Whitaker. We will see some of the characteristic features regarding this Monera. They are unicellular organisms. Unicellular organisms means their body is completely made up of one single cell, but it is carrying on all types of metabolic activities that is required for the bacteria. Then, why it is called as prokaryote? Monerans are prokaryotes. What are prokaryotes? Prokaryotes means the organisms that does not have true nuclear membrane. All the bacteria which does not have nuclear membrane around their genetic material are called as prokaryotes. Their cell wall is made of peptidoglycan. Now here I have used the term cell wall. Cell wall means outermost layer of the body that is made up of carbohydrates only. In addition to carbohydrates, there may be other biomolecules, but the major content should be of carbohydrate. Then that outermost layer is called as cell wall. Next one, cell wall is made up of peptidoglycan. Now, what is peptidoglycan composed of? It is composed of N-acetyl-nuramic acid plus N-acetyl-glucosamine. N-acetyl-nuramic acid. NAG, N-acetyl-glucosamine. Peptidoglycan is covered on its outer layer by Glycocalyx. So this is the outermost layer that is present on the cell wall. Now, Glycocalyx is of two types. First type, if the layer is loosely bound to cell wall and is irregular, then called as slimy layer. Next, Glycocalyx the layer is tightly bound to cell wall and is hard, then called as capsule. To know all these labelings, we should know about diagram of bacteria. Let's label this one. This is cell wall of bacteria. Innermost one is plasma membrane. These are called as mesosomes. This is 70S ribosomes. This is DNA and this is gelatinous material. Outermost layer is Glycocalyx. This Glycocalyx can be of slime layer or it can be of capsule. All this will be cytoplasm. Here I did not mention about flagellum, sexpili or fimbriae. Just we have represented some parts of that. The next coming points we will represent where are those structures. Bacteria or a Monera does not have membrane bound cell organelles. What are membrane bound cell organelles? Golgi complex, endoplasmic reticulum, mitochondria, lysosomes, etc. Like Golgi complex, endoplasmic reticulum, mitochondria, lysosomes. All these types of cell organelles are absent. The type of ribosome is 70S ribosome. Then first doubt, what is meant by 70S? These ribosomes, when they are centrifuged and then allowed for settlement, then they are separated because it has two different bits. One is 50S, larger subunit, and smaller subunit. These both are separated according to their concentration gradient. So basing on that, different types of ribosomes are given. Eukaryotes will be having both 70S and 80S. Prokaryotes will have only 70S type of ribosomes. S indicates Swedberg unit, basing on the sedimentation of the larger subunit or the smaller subunit. Ribosomes are of 70S. Next, plasma membrane is thrown into inner foldings called as mesosomes. So here we have represented mesosomes. Here we have written mitochondria are absent. Mitochondria in case of eukaryotic organisms are responsible for providing ATP and also carrying the respiratory functions. So it has many number of respiratory enzymes which provides ATP and also oxygen if necessary for the organism it will provide. But in case of bacteria, what is the structure that is responsible for respiration? Mesosomes are the structures which are equal to that of mitochondria, but these are mitochondria are not present in prokaryotes. So mesosomes are existed as the respiratory structures in case of prokaryotes. These are equal to mitochondria of eukaryotes. Coming to DNA. DNA is circular, naked, double-stranded DNA. So we have written two number of strands. So circular, naked, double-stranded DNA. Again, prokaryotic DNA is the oldest DNA. So called as incipient DNA. The oldest DNA is termed as incipient DNA. Next, prokaryotic DNA is a thread-like, circular thread-like, so called as fibrillar DNA. Now, in case of bacteria, DNA is not covered by nuclear membrane. So, there is no nuclear membrane, but DNA is present in gelatinous material. Now, we will give a name for that. DNA plus gelatinous or mucousy material is combinedly called as nucleoid. So, in case of bacteria, DNA plus the gelatinous material around the DNA together are called as nucleoid. Mesosomes are the structures that are equal to mitochondria of eukaryotes and responsible for respiratory functions. Now, when DNA want to undergo the division, like binary fission or multiple fission, then for availability of ATP, it goes near to the mesosomes and then starts division from there. Nucleoid, also called as genopore. Now, bacteria, we'll write some bacteria have flagella or flagellum for locomotion. So if this is a motile one, like this, the flagella are long structures. Flagella or flagellum made of protein flagellin. Bacteria has pili, structures that are used during sexual reproduction. So pili are very small or shorter one comparing to that of flagellum. They help in adhering to the other type of bacteria. Here sexual reproduction in the sense, basically gametes are absent here, but sexual reproduction means if one bacteria is having special type of genetic material, that is fertility factor, and other bacteria is not having any fertility factor, then in the later stage, if this fertility factor want to enter into this bacteria and make it adopt for the newly changed atmospheric conditions or environmental conditions, then it will give a Xerox copy of this. Later, these both will form a connection tube. Then this newly formed one will enter into the fertility factor absent bacteria. This process of transferring of fertility factor is called as transduction. We call this commonly as sexual reproduction in bacteria. So pili are the structures that support during sexual reproduction by adhering to the opposite bacteria. So they are also called as sex pili. This sexual reproduction in bacteria, also called as transduction or bacterial conjugation. There are some structures that are very much long comparing to that of flagellum, longer than flagellum we can say. So these are the structures that help the bacteria to adhere to the substratum. Fimbriae are longer structures. They are also made up of flagellin only, that help bacteria to adhere to substratum so that it can colonize later. Bacteria does not have histone proteins which are present within the coiled forms of DNA in case of eukaryotes. Those histone proteins are absent in case of prokaryotes. Basically, reproduction is by binary fission. Mode of nutrition is autotrophic, that is seen in case of eubacteria, and also chemotrophic bacteria. Here under autotrophs we have two types. By using light are photoautotrophs. By using chemicals are chemoautotrophs. Some bacteria depend on other organisms for food material. So mode of nutrition, one is autotrophic, second one heterotrophic or heterotrophs. Some bacteria also help in degradation of the dead particles or dead organic matter, that is saprophytic. So here we have used the term heterotrophs, saprophytic and autotrophic. So three types of nutrition, and some bacteria are parasitic. We'll write this point separately. Some bacteria cause infections on other organisms or to other organisms. Are called as parasites. Next, in bacteria we have two different types of bacteria based on the oldest type and the newly originated. Oldest one are called as Archaea Bacteria. The new one means which later modified themselves are nothing but Eubacteria. Under Eubacteria, we read the example like cyanobacteria or blue-green algae. Bacteria based on evolutionary time are of two types. One is Archaea Bacteria. Second one, Eubacteria. Archaea Bacteria are kept in sixth kingdom by Carl Woese. So here, Archaea Bacteria cell wall is made of N-acetyl pseudomuramic acid. Next, N-acetyl glucosamine. Glucosamine is common in case of Eubacteria and this one. But in case of muramic acid, here the cell wall is made up of N-acetyl glucosamine, N-acetyl muramic acid. What is this pseudomuramic acid chemically called? Chemically, it is talosamine-uronic acid. We call this one as pseudomuramic acid. This one is also written in shortcut as N-acetyl T also. This Archaea bacteria has branched lipids. So, it is able to tolerate extreme conditions. So, under Archaea bacteria, we have types are thermophils, second one halophils, next one methanogens. Thermophils can tolerate high temperatures, means even above 120 degrees centigrade also. Halophils can tolerate high or normal salinity, means salt conditions. Salinity. If water is having more minerals or salts, even then Archaea bacteria can survive there. Methanogens can tolerate extreme anaerobic conditions. Next, under eubacteria, example is cyanobacteria or blue-green algae. For examples of bacteria are Mycoplasma, Rickettsia. Next, Pseudomonas, Streptococcus, Clostridium, Actinomycetes. It is gram-positive bacteria. It is also called as ray fungi. The smallest bacteria is Dialister pneumosynthus. Largest bacteria is Thiomargarita namibiensis. The smallest living organism, so its measurement is 1 to 6 micrometers. It is 700 micrometers. The smallest living organism is PPLO or Mycoplasma. PPLO full form is Pleuro pneumonia like organism. Mycoplasma is not having cell wall. So the bacteria without cell wall is Mycoplasma. Basic size of a circular or a spherical bacteria is 0.5 to 2 micrometers in measurement. This is called as size. But a filamentous bacteria has 1 to 10 micrometers length and 0.25 to 1 micrometers in diameter. These are considered as the measurements of bacteria. Based on shape, bacteria has different shapes. Are of first one, circular shape called as coccus or cocci. This is round one. Round one, single one is called as monococci. Coccus. If always two bacteria which are circular in shape are existing, then it is called as Diplococci. If One single one is monococcus, that is singular form. And if two cocci are present always in the same place, then those are nothing but diplococci. So diplococci are example Streptococcus species. Now these are studied by Griffith, and this term is given by Griffith when he started studying on rough and smooth strains of Streptococcus species. Second one, rod shaped, is called as bacilli. This is in this form. Third one, bacteria in grapes bunch like, then it is called as Staphylococci. Next, in cube form, that is bacteria always existed in a cube form like this. We can imagine at each corner one bacteria, means those bacteria are living in a colony form. Sarcina. Next, fifth one, if cocci or bacilli arranged in a long chain like, then term is Streptococci. Next, if comma shaped, then called as Vibrio cholera or Vibrio. Example is Vibrio cholera bacteria, which causes cholera. Next, another bacteria is spirally or spiral. Then example is Spirilla or Rhodospirilla. Spirilla. Its shape is like this. Like this. So, based on presence or absence of flagellum. First one, A. trichus, no flagellum. Example, lactobacilli, which is present in milk. Bacilli means rod shape. Second one, monotrichus, one flagellum. Example, Vibrio cholera. Third one, Amphitrichus, one flagella on opposite side of body. So here we can simply write this one lactobacillus as like this, comma shaped like this. In case of this structure, we can draw the diagram of this one single flagellum on either side. Example is Rhodospirillum. Fourth one is Cephalotrypus. We can use a term one bunch or one tuft or one or many flagella on one side of body. Example can be Pseudomonas fluorescens. Next one, Lophotrichus. Tuft of flagella on either opposite sides of body. Here we can frame diagram like this. Here it is tuft on either side. Example is Spirilla. Last one is Peritrichus. Flagella all over body. We can take, if this is the bacteria, all over the body like this. Example, E. coli, Escherichia coli.